It's recording. Okay. All right. So welcome back, everyone. Today, the plan, we're going to take a little deviation from the Strogatz book and look into possibly the greatest mathematical model in all of biology. And in my opinion, one of the greatest sort of scientific achievements ever. But interestingly, sort of overshadowed because of the time that it happened. The Hodgkin Huxley papers came out in 1952, late 1952. Can anyone guess what overshadowed it a few months later?
DNA.
Yeah. So yeah, and rightly so. The implications of that were huge. But so I've been I took a deep dive into the history of electrophysiology over the past couple of weeks because I wanted to make sure I understood a few of the concepts. And this one book I was reading called Galvani's Spark, which I highly recommend. I'm sort of in the middle of it. Gets into this why the DNA discovery is kind of more stupendous in a way. But interestingly, the period in which people discovered that it was deoxyribonucleic acid and not proteins that were the carriers of heredity was a decade or less or something. So it was a relatively short period of time between identifying DNA and discovering what it looks like and how it works. Whereas the story of electrophysiology, as we will see, is something like 200 years, to 150 years up to Hodgkin and Huxley, roughly 160 odd years. And it took a while. There were moments when it could have been sped up, but that's how these things go. So just to recap what we talked about before, two parts of the making of a dynamical system, and I'm only going to really talk about the first two, is identifying some key forces and turning them into dynamical intuition. Fixed points and linearization we'll probably only talk about next time for this system. But let's kind of start right from the beginning of electricity. Luigi Galvani, after whom the galvanometer is named, discovered animal electricity in 1780. So he was working with his wife in the University of Bologna, the first sort of modern university in Europe. And he sometimes described that he accidentally discovered this, or it was a chance discovery. But as I have discovered in the past couple of weeks, that's sort of unfair to him because he was quite prepared in a way. And the line came up, fortune favors the prepared mind, which is very true of him because he was doing several experiments to do with muscles and but also to do with static electricity. So this apparatus here you can see in the middle, that big round thing. Can anyone guess what that is? The thing with the crank, the lever thing. This.
I assume that's for generating static electricity.
Yes. I believe they just called it that. I forget what, but yeah, it might have been called a generator. So he was, I think what happened was a scalpel he was using picked up some of the charge he was generating, and then when he touched the exposed nerve of the frog, it twitched. And it was a momentous twitch because Alessandro Volta, who was one of the early physicists of this time, he was quite famous already, and he was intrigued by this finding which Galvani published, and he started to look into this. And over the course of this biological discovery, he invented the voltaic pile, the first battery. But he also disagreed with Galvani. Galvani thought that the electricity came from, or the twitch was caused by the electricity, and that electricity was a sort of natural property of the nerve fiber. But Volta disagreed, and it was a weird sort of historical quirk that he discovered that bimetallic strips could produce a current, and Galvani himself had observed that and was using that to cause the twitch. And so Volta said that Galvani was wrong and that it was some other reason that the twitch was happening, and that the current came from the metals that were being used. So for a while, people were a little bit confused about what Galvani had actually discovered. And even in modern histories, people often with the sort of physicist bias say that Volta corrected mistakes on the part of Galvani. But as it turns out, Galvani was right about the presence of electricity as such in nerve fibers without the need for bimetallic strips or anything like that to produce it. So, what is electricity? Would anyone like to? I mean, there's a standard definition, but
I'm gonna guess it's the flow of electrons from one point to another. Is that it?
yeah, that's a start. That's basically it. But as we'll see in the case of animal electricity, it doesn't have to be electrons only. It can be charged ions, and that's the broad concept. But when we talk about it, we have all these other terms, right? There's power, current, voltage, charge. What are all these things? So charge, I think people are pretty familiar with because it's a sort of atomic property, and people are aware of electrostatic forces of repulsion and attraction. Current is the flow of charge. What exactly is voltage? This was the thing that always used to kind of mess with me, because I would be able to solve the problems in physics class, but I was like, what is it? So what is voltage? Anyone? Venture a definition.
It's potential. It's like a potential energy.
Yeah. It doesn't have the units of energy, but that's like a sort of semantic thing. But yeah, that's basically what it is. So it's the potential for the flow. And for that reason, in fact, I discovered this hydraulic analogy of electricity only in the past couple of years. And it definitely breaks down eventually. But I think it's a really useful way to get started with understanding electrical circuits. And then you sort of correct it along the way to show why it breaks down. Because otherwise it can be quite abstract, understanding these invisible flows. So there is a reasonably systematic analogy to be made between the flow of water in pipes and the flow of current, where the volume of water is analogous to charge, the flow rate of water is analogous to the current. The pressure, and that's the one I wasn't so clear on, pressure is analogous to voltage. Energy is the same, power is the same, basic concept. So so you can imagine, when pipes get more narrow, there's resistance. There's you can even make equivalents of diodes and transistors using this analogy. A transistor basically allows a weak current to control the flow of a strong current, and so you can imagine a weak flow of water controlling something that directs and the flow of a much more powerful stream of water. So if anyone has any sort of even very basic questions about flow of electricity and potential and electromotive force, this is a good time to ask, because I found that even though I took physics, I was a little unclear for years in grad school.
Is voltage analogous to different levels of water in different locations that level out?
Right. You need quite a bit of flow for that. So you're maintaining an energy difference, say with height, and that height is what causes the pressure of, say, of some water.
So in the water analogy, is the difference in height proportionate to the voltage?
I'm not sure how the numbers exactly.
That would be like a
work out.
voltage difference.
Yeah, the voltage difference is what you do. Yeah.
You could think about a pipe as like a pipe connecting those two. Before they're connected, nothing happens, right? These two tanks. But then when you connect them with a pipe, you create a flow and they equilibrate. And that would be like having two charges, two charged spheres, right? Connecting them by a wire. And then, well,
Um, I
okay, maybe
mean,
that's not right because I was thinking about charge. I want to think about voltage this way.
so is what I'm envisioning is that this is a capacitor for water. Like you've made a water battery where there is a potential of a potential. And then once you remove or make a hole in the wall, then you have an actual voltage.
Yeah, that kind of makes more sense in a way because in the container analogy, you have the water levels equilibrate. But if you had two batteries, then you would have a continuous current and it would flow until your battery ran out.
So in the case of the capacitor, you're charging it up, and so this is a really good set of analogies because with the capacitor, you gradually lose the current if you do connect something, because the capacitive current means the discharge happens. So what capacitance is, if anyone is confused, is the accumulation of charge on two sides of an insulator. If it's a perfect insulator, then it's a perfect capacitor. And you need energy to maintain that difference. Otherwise, typically, well, in the capacitor case, not exactly, but in the case of flow of water, you need to maintain water pressure to maintain a constant flow of water through pipes. If the water level continuously decreases, that's a little bit like the capacitor discharging.
Mm-hmm.
So,
yeah, or the battery running out or something.
Right, or a battery running out. So in the period when the battery is like way is well charged and not near running out, it's more like this constant flow, which is not like a capacitor. So and is like a cell, is like a battery. So constant pressure, and an interesting thing about that is that that gives you the picture of this as a driven system, meaning that you have to supply energy from outside. In the case of water, there's energy coming from streams or geological forces, and climatic forces are producing that energy or transferring it to where you need it. And in the case of the battery, you stored up a lot of chemical energy in the home of the battery. So that's good to clarify these things before going into some of the details. Any other doubts, question about electricity? Because I always learn something new when this topic comes up.
So
Anyway,
it's correct to think of a battery as like one chamber that's full of electrons and another chamber that's lacking electrons. And when the battery sort of runs out, then they're equilibrated.
That seems reasonable.
more or less,
Sort of have
although
the
you're
anode and
probably
the
aware of the actual mechanism involving salt and things like that, right? So so yeah, I think that's a reasonable way to think about it, that you've kind of like stacked up a lot of water, like a quite large amount of water, and arranged it so that, and you're using a very, very small pipe come to at the bottom of it, so that for large periods of time you get a relatively constant flow. So basically the pressure loss is small compared to the time scale. The time scale of the pressure loss is small compared to the time at which you're interested in the outflow, because what you want is a constant, relatively constant current in the case of DC direct current. Okay, good question. Good point. So jumping back to the history, Galvani, I mean, despite Volta kind of causing a little bit of controversy and seemingly basically kind of dismissing what Galvani had done for a while, by the 19th century, you start to see people rediscovering some of these ideas and putting them on more firm experimental footing. So Matteucci followed up on Galvani's ideas and showed that there is in fact a real potential difference between a membrane, or the inside of a membrane, and the surrounding. And by this time, basically in a relatively short period of time after Volta, you have all the devices needed to do the measuring, the ammeters and voltmeters and all of that. So after Matteucci, I think Emil du Bois-Reymond, despite the name, he's a German. I think he saw Matteucci's book or yeah, I think he saw the book, and then he decided to look into this also in Germany and discovered the action potential. I don't think he had any recording technology for it, but he saw basically this deflection in this steady potential difference. So basically, with a cell that's still healthy and not very damaged, there's a sort of constant potential difference maintained in the quiescent state, in around 55 to 65 in a lot of minus 55 to 65 millivolts, meaning that the inside of a living cell is at a lower potential than the outside. So Bois-Reymond taught a lot of important physiologists. This is around the time that physiology kind of was born as a field. It's the much younger brother of or sister of anatomy, which has been around forever almost. But physiology is new. So Bois-Reymond's student Bernstein made this first somewhat accurate recording of an action potential, and the way he did it was truly brilliant. He made this device called a rheotome. If any etymology fans can explain what rheotome means. It's the same tome in microtome and tomography. So basically, and the word rheo means flow. So it's a time slicer. So what he did was, at a time when most when the ammeters and galvanometers were way too slow in their response to record something as fast as an action potential, he used this rotating circuit breaker cum recording system that effectively was able to sample the action potential at sub-millisecond sub-millisecond resolution. It's quite amazing. So it's worth looking into that if you're curious about 19th century mechanical engineering-based kind of creation of apparatus. So if you look at B, that's voltage versus time. But you may notice something odd about this. People who've seen action potentials will notice something slightly weird about it.
Why is it going down?
Yes, that's part of it. But if I tell you that could be a convention issue, is there anything else that's missing? So what he's actually plotted is only the after hyperpolarization. He didn't actually show the spiky part of the spike. Apparently he observed it, but didn't believe it was real. So this is a very interesting aspect. So when we think of the spike, we have a very specific shape in mind, which I'm assuming all of you have seen. But when people talked about the nerve impulse, that very sharp part of it was initially it wasn't clear why that would exist, and it was very non-ohmic. So people in Bernstein's case, because he lived to see the recordings that included it, he regretted not reporting it because he was under an assumption that the potential couldn't reverse and go in the opposite direction, basically. And there was a debate about what exactly was causing it. So he left it out of his plot, even though he saw it. So another concept which was circulating at the time was, well, what is causing this potential difference? We talked about electrons moving as being part of electricity, but in this case, it isn't electrons. And how, I'm sure a lot of you are familiar with where we're headed. So, we'll talk about how sodium and potassium ions are involved in this story. Interestingly, important constituents of seawater. So I like to think that the action potential is, in some sense, a dynamic involving things that have been with life since, the primordial sea. But anyway, when I was looking into the story of the dynamics of Hodgkin Huxley, I was thinking, well, how did anyone measure all of this movement of sodium and potassium? Because they already knew about that, or at least they knew qualitatively that sodium and potassium were involved. So the question is how? And what I uncovered just in the last week was that this person, Ernest Overton, who is still neglected in a lot of these histories of neuroscience, played a really important role because he studied osmotic pressure. Does anyone know what osmotic? I mean, I'm sure a lot of you do know what osmotic pressure is, but would anyone like to define what it is?
Just difference in concentration of ions. Is that
Right, right. And that's what causes it, basically. So he observed this, and this is something that started out in plant physiology, actually. And he was looking into this, and then he started using the same plant methods in animals. And he looked at different solutions outside the cell and found that, and he basically was on the right track talking about sodium and potassium exchange in the late 1800s. And I think Huxley may not even have been aware of him originally, but he said that if people had been paying attention to what Overton had done, he was in Sweden at the time and not a very prolific, he wasn't good at publishing his work. He did good work, but was kind of not interested in the publish or perish business. So yeah, he said, I think Huxley is exaggerating but he says that, his work with Hodgkin would have been obsolete if people had followed what Overton was doing. I think there would have been experimental challenges to prevent people from fully capitalizing on Overton's insight, but they could probably have done it by the 20s or 30s if they had been paying attention. Anyway, so a little bit of an aside to this historical aside is like, what is osmotic pressure? So we jump back there to again to the 1700s to Nollet, I believe, who first observed it in plants, and then Wilhelm Pfeffer, who made the Pfeffer cell, which you see here, which was a way of measuring it involving a column of mercury. But it's easier to look at this diagram up here, where this bit in the middle of the U is a membrane that's been placed there. It could be various things, semi-permeable membrane. And initially you have just pure water on either side, and you can put a large, it doesn't have to be that large, it's molecules or ions or salts in one side, and you see the water flowing from the region of less concentration to the region of more concentration. And this is something that we sort of all experience, just in dealing with water and solutions. And I'm sure everyone has heard these terms: hypertonic, isotonic, and hypotonic. These are just blood cells. So if you're just looking at cells under a microscope and treat them to different solutions externally, you'll notice that sometimes they shrivel up. And if they shrivel up, they get damaged. So this is actually an interesting technical challenge, which people in the early 20th century, including Hodgkin and Huxley, solved in various ways with isotonic solutions. So if they wanted to do experiments where they reduce or increase the level of sodium or potassium, they had to do that without changing the osmotic pressure. And that involved using things like dextrose and a couple of other chemicals, which were not charged and therefore didn't interfere with the electrical properties that they were interested in. It's quite a fascinating set of things that had to all sort of be lined up in a row for any of this to have happened. So around this time, Americans start to really make major contributions to electrophysiology. Kenneth S Cole, I think, was one of the first to use the then new cathode ray oscilloscope, which was the only way to plot something this fast accurately. So just imagine when you have sort of relatively slow photographic paper and like mechanical things involving pens. It's not very easy to record something that is like 3-4 milliseconds in duration. So they managed to do it. I'm not sure how they transferred the image from the CRT to photograph, but something to look into. And here, this plot, which I had seen before but never really knew what it was, is a combination of an action potential on top and a measurement of the impedance of a neuron during an action potential. The reason it's two-sided has something to do with the way the oscilloscope measures impedance, which I didn't fully understand. So they showed that during the action potential, the impedance suddenly changes. So apparently this image was a hit as modern art in Sweden, and was in this vertical form, apparently showed up in apartments. So yes, what is an action potential? You've all seen it, probably. What would anyone like to define it? Well, it's clearly a movement in voltage, but where? So one thing that I've noticed a lot of people kind of get confused about is the direction of flow of ions that are involved in this deflection of current. Is that because the first thing you might think when you look at these long axons is that something is being sent from the cell body all the way down the axon. And that's not really what's happening. The main flow of ions that produces this change in a potential is across the membrane, meaning sort of perpendicular to the direction that the axon is going. So you have these little local circuits of flow across the membrane, and they each excite neighboring parts of the membrane in a sequence. And this idea, amazingly, Bernstein himself was able to put forward more or less this idea that
Ask a question.
this.
So the zero in those plots of the voltage, that's the relative to the extracellular medium?
Yes. Well, you have to watch out with some of these plots. I think this one has been corrected, but they used to use a different convention for inward and outward flow, and so it's a little bit confusing. Basically, the way that they labeled what the ground was changed after the Hodgkin Huxley papers were published. So the link I sent was to this updated translated version, which uses the conventions that everyone uses now. But this looks more or less like the modern convention. So maybe the British were not using, maybe the Americans had already moved to the convention we use now. Well, I'm not sure why this might be corrected, but yeah, the squid giant axon, which is what they measured from, was around -65 millivolts with respect to the outside.
So that's a voltage drop across the membrane measured at a point on the axon over time. Is that right?
Over time, yeah. So it's not the spatial flow, which looks similar, and which Hodgkin and Huxley also modeled amazingly. So yeah, so this started in 1939, just before the war broke out, and that caused a major delay. So a lot of this could have been done by 1945 if the war hadn't happened. But they were actually thinking a lot about this problem while the war was going on. So in this diagram, you can see the basic idea that was already circulating in Bernstein's head and was popular. There's some interesting quirks here about when you cut an intact cell, there's an outward flow of current, which was also in need of being explained. So for a while, this current that was measured was called an injury current, because they thought that some people thought that it only occurred when there was an injury to the cell. So that's a bit of so, and in C, this is what I was talking about, local currents flowing they call them circuits, which originally confused me when I was reading the Brown guide. So these loops going across the membrane and back were called circuits in the old days. So basically that was the way to explain how something could travel along the axon despite current flows perpendicular to the axon, more or less. And this GIF shows the basic idea. Does it work for you? I don't know. It's on the Wikipedia page for action potential, and I like it a lot. So that shows this what's happening spatially also, more or less. So, Hodgkin and Huxley, these are the players in this whole story, and also Bernard Katz, who worked with them. Unfortunately, he wasn't also awarded the Nobel Prize. I guess they really liked the mathematical part in addition to the experiments, and only Hodgkin and Huxley worked on that part. But I always feel sad for when people get left out of these prizes. It just makes me not like prizes. But if anyone was deserving of prizes, it's this group here.
Would in that first picture, I feel like I've seen this picture before and I confused. Not this one, the next, just the Hodgkin Huxley picture. Is that a squid?
No, that's some sort of lobster or something.
Oh, that's not even
Which I found funny. I think I've seen this before, and I found this picture funny that time too.
It's pretty hilarious, yeah. Yes, it's not a squid. That is the cathode ray oscilloscope, though. And this must be from the 60s. When they were doing this work in the 40s and early 50s, they were like, in the 40s, the 30s, they probably looked like this. Yeah, that's some sort of lobster.
Yeah, so because invertebrates don't have myelin, their neurons tend to be much larger and thicker, especially along the spinal area. So that's why it's easier to do neuroscience, this kind of neuroscience on invertebrates. It's just you have more room to work. So lobster works, squid works, any sufficiently large invertebrate works for this.
Yeah, so there's a major difficulty in recording inside the axon, which is the size of axons in most of the creatures that one would encounter. So this is an electron microscope image comparing a rabbit nerve to the squid giant axon.
Because they are very big, one of the innovations that they could do then was to just use a glass contact and directly pierce and basically do like a version of a patch clamp, but it was very big because these neurons were long and they have huge diameters and so on. You can't do that with rabbits and rats and monkeys and whatnot. So it actually helped that if you look at the electrode contact, in fact, if the picture that shows the lobster, where when I was in grad school with Jen Wei Lin, we actually did experimental work with crayfish, and we were to draw our own glass electrodes, and they were big enough that we could draw it on our own from the lathe, whatever the equivalent of lathe is, so you can actually make your own pipes to actually hold the sodium ion or whatever ion you're interested in, and you can actually like pierce through the neuron without damaging it enough and record from it simultaneously. So it's also a great luck or accident that they also pounced upon the right animal model to actually come up with a measure of the electrical activity. That's a very important achievement as well.
Yeah, because the recording electrodes were just not big enough to penetrate. You could see this, you could do this with the naked eye, a lot of this. So yeah, there's these two giant axons. So it's not a giant squid. it's only around this big. It's the axon that's giant. So it's not, yeah, saw some other lecture. It's not the kraken. So this guide is really useful, although it's sometimes easier to just read the original paper than his guide, but occasionally it helps because they did a lot of work It's really an experimental and theoretical tour de force. No other way to put it. There's just so much that they ended up recording and inferring, and the clever use of math that I couldn't really summarize all of it because it would probably take a lot of time, and it's easier to just read it on your own. But some highlights that I just pulled out. So from Brown's guide, he just says it is the first mathematical description of biological phenomena summarized in an empirical model of impressive predictive power that successfully reproduced the action potential. And you could quibble and say that there were other, mathematical descriptions, Darcy, Thompson, and all that. But so maybe if you say description of a process in time, then this is a pretty accurate statement. So this is the model. I'm just dumping the equations on you. Since we've already looked at equations, you should at least know broadly what this is. And you'll note that it's 4 dimensions. And it's based on basically the standard way of treating currents, voltages, and resistances, and capacitances. So it's just the sum of a capacitive current, a potassium current, a sodium current, and a leak current, which involves chloride ions. This is a passive current, which just sort of leaks out. And these 3 variables add up so that you have, in addition to voltage, it's a 4-dimensional system. The N, M, and H, deriving the properties of these entities is the main contribution of this paper. And when this is often taught to people, they talk about ion channels. But Hodgkin actually didn't know about ion channels. Ion channels hadn't been discovered yet. So they postulated that you could think about these flows in terms of conductances which were active and voltage dependent. And they showed that they were voltage dependent using these very careful experiments. And so the first equation here can be understood in terms of an equivalent circuit. So each of the active conductances is connected to a battery, which is the reversal potential of that particular ion, which is the potential at which the ion's flow ceases. And then if you keep changing the potential, it starts to flow in the opposite direction. That's what reversal potential means. And they're not standard resistors because they behave in quite wild ways compared to ohmic resistors. And there's a capacitance, and there's a capacitance associated with the membrane, which roughly means that even though ions are able to flow, there's always an accumulation of ions on either side, sodium and potassium, that maintain the potential. So it's very good that we talked about that capacitance energy bucket of water business, because if all the ions flowed, then you would not have any maintenance of this resting potential over time. It temporarily breaks and goes in the opposite direction, but the capacitance sort of is one of the ways to understand how that's maintained. But there's also an active component, which is ion pumps, which again were not known at the time, to basically restore balance after an action potential has occurred. So what did they do? This diagram really it's almost self-evident what they did, but so in the early paper with Katz in 49, Hodgkin and Katz, they showed that at rest, the membrane was predominantly but not exclusively permeable to potassium. These experiments are quite tricky because the cell gets damaged much more easily when you change potassium concentrations than sodium concentrations. But they managed to show that by changing the external concentrations of sodium, you could alter the resting potential. And the other ions didn't matter. A little quirk here for people who are fans of mathematical tricks is that the way in which they were able to quantify some of this was that they used the Nernst equation, which is an equation which is a logarithmic equation relating the voltage across a semi-permeable membrane with charged ions with the concentrations on either side. And the thing is that it was virtually impossible to record the concentration of ions inside the cell. How would you do that even? So because the Nernst equation is logarithmic, there's a trick you can do where only the manipulations on the outside matter, where basically you subtract the two different external readings and the internal concentration gets canceled out. If you've seen the Nernst equation, you probably know what I mean, but basically it's using the property of the logarithm. So these are experimentalists, but also people who are very aware of mathematical properties, and that enabled them to design experiments very well. So knowing what could be canceled out by virtue of the property of logarithms helped them to just use external manipulations of ion concentrations to get what they wanted. So they didn't even have to measure the concentration on the inside, despite the fact that it was needed in order to calculate from first principles what the voltage was. So they were able to show that that's what potassium did. And this figure here shows that the action potential, which is not as opposed to the resting membrane potential, depends primarily on sodium influx. So sodium rushes into the cell briefly. And they did that by systematically lowering and then raising back up the concentration of salt in the external solution in which the axon is kept. So this these numbers are the order in which they did this to the same axon. So 1, 2, 3, 4, 5, 6, 7, 8. And they've basically killed the action potential, despite the fact that they've stimulated. And then they go back up again, 9, 10, showing that it's reversible. Yeah, so everything they've done show extreme care. So then in the 50s, with the series that was published in 52, they started using this new technique called the voltage clamp, which was developed by Cole and one of his colleagues. And so they were able now to, instead of just sort of passively watching what the cell does in response to a stimulation, they could control the voltage and then see what the cell does. So this gives you much more exquisite control over what's happening. So you can separately fix what the voltage is and find all the voltage dependent currents. So it helps to keep track of these terms, which are, I think, initially can sound quite confusing. So would anyone like to just go through them? What is depolarization? What is polarization? Why is it called polarization first?
Okay, so the resting membrane voltage is negative. Depolarization is when it goes towards 0, so the voltage goes up because positive charge is coming in. Hyperpolarization is when it gets below the resting voltage, goes negative because negative charge is coming in or positive charge is leaving, depending on how you look at it. So yeah, so inward current is just anything where inward current is positive charge flowing into the cell, correct? And then outward current is negative charge, is positive charge leaving the cell.
Yeah.
Okay.
So when I first encountered these terms, I was very confused because if you think about it, the cell is also polarized when at the peak of the action potential. But it's just a convention now. It's just the opposite polarization. But that's just the convention now, that when you further make the membrane potential lower, then that's hyperpolarization, and when the other direction is depolarization. Inward current, an interesting thing there is, I'm sure a lot of you are aware of the arbitrariness of the convention of positive and negative charges. And some people think, well, if it wasn't for Benjamin Franklin, we would have had much less confusion in school kids because the direction in which electrons flow would be the direction in which the current is positive. But for electrophysiologists, it works out great because we're dealing with positive ions. So, so thank you to Benjamin Franklin for that quirk of arbitrary conventions. Okay, so in the first of the 52 papers, submitted in 51, I think, Hodgkin, Huxley, and Katz basically studied voltage-dependent currents, and so what does the voltage clamp do? You're holding the membrane at a particular voltage, so the voltage can't change. So the only thing that can change is the currents, and the reason the currents change, as they show basically, is because of changes in conductance. So what you can see here is that when in A you can barely see it, but they give a small hyperpolarization, and you basically see something like a relaxation back to resting. It's very small. They zoom in on this later. But then when you see, you can barely even see it because they're using sparks here rather than constant current injection, approximately. So they have a slight depolarization. And so remember, we're looking at current here, or current density. You see this, and in this convention, down means the inward current. So going in this way and then going out here. So in is down. And this bump here is the main kind of, if you look at the time scale, you can also kind of figure it out that this bump in current seems to have something to do with the action potential. Here on the right, you see the capacitive part zoomed in, basically what happens right here. So if there isn't any possibility of the sodium current flowing, you see a symmetrical, more or less symmetrical capacitive current. So that just shows that that part of the system works more or less how you'd expect a near perfect capacitor to work. Symmetrical on both sides, relaxing back to normal. Next, there's each of these papers has a few other things, and I'm just sort of picking highlights. But next, what they looked at was the sodium current in more detail, and they use the same technique, but now with the voltage clamp, which is changing the concentrations of sodium and just measuring everything that they could possibly measure. So up here, you see with the normal amount of sodium, you have this inward current, and then this later outward current. And when they basically knock out all the sodium, that bump is gone, and instead you see this other sort of shape. Here's it, and it's not just based on the presence of absence of sodium. The specific voltage matters for the time course and the shape of the current. So they carefully looked at how that shape changes for both the sodium and the potassium conductance. Even here, a lot of mathematical cleverness came in, because what they're able to record is just the overall current. There's like, how would you in principle separately record the potassium current and the sodium current? This is years before the patch clamp, and in any case, even in the patch clamp, I think you get both sorts of ions can flow. So they used their salt manipulation methods and some very clever mathematical tricks to subtract out basically two components from this overall plot, so that they could kind of factorize this highly nonlinear plot into a sodium current and a potassium current. And again, so I see I've skipped over one of the middle papers, but then right before the modeling paper, this paper showed a very crucial kind of property of the cell, which is inactivation, which is that sodium permeability increases, which produces that little bump initially, but then something brings it back down, which is the other side of that bump, right? And that's also voltage dependent. And they showed what that voltage. So they think of this a theoretical concept which they introduced as something which, when it's 0, sodium can no longer flow. When it's 1, when it's 1, sodium can flow freely. And this depends on voltage. So we're carefully able to use changes in the size of the action potential, of the current, to infer how that gating. I don't think they use the word gating, but nowadays we call it the gating of the sodium current, or nowadays we say gating on the channel. So back again to the model, which they so they basically had to fit lots and lots of complicated data with that had to do with the time varying aspect and the voltage varying aspect. They could have done it in various ways, but they used typically exponentials of various sorts, and they got and then they cranked out these plots. Now, one of these two plots is the model. Can anyone guess which one it is? It's virtually impossible. The top one is the model.
Yep. Go for it.
What they're really good at, it's just amazing, is how carefully they look at all the discrepancies between the model and the real measured action potentials. They look at how temperature influences the action potential. They look at all these little deviations in shape, and they try to explain all of them and why the model fails to capture every little part of it. So here's another.
Just a question about just the methodology here. I mean, I didn't read the paper directly, but how are computers, like, did they have this? There are so many variables in this equation. How did they fit anything?
I have a slide specifically about this.
Excellent, thank you.
You will see in just a sec. Now, I didn't really pause to talk about it, but you may have noticed that the gating variables on the sodium and potassium involve these functions n, m, and h raised to some power, right? N is raised to 4, m is raised to 3. So when I first taught the Hodgkin Huxley model, I was like, where on earth did these powers come from? Like, who would think of raising an exponential function, itself complicated, to the 4th power? And here the fastidiousness of Huxley really comes in, because now this plot here, which is in Brown's guide, I don't know why the pictures were so low res. You see this 1 minus exponential function for n, which is the potassium opening variable, which is much slower than the potassium one. Sorry, much slower than the sodium one. And here's n to the 4. If you were like me, you would think, well, qualitatively the same. But basically, they noticed that there's a little bit of a knee point here, a little bump in the onset of the potassium current, which they wanted to model. And they could have done it in various ways. Nowadays, people might use polynomials, and you know, there's all kinds of ad hoc ways of doing it. But based on sort of kinetic theory and Boltzmann-style reasoning, they worked with these exponentials, but this power of 4 didn't come from there. Huxley, just in a moment of brilliance, which, you know, you can't really say what the method was, he reasoned that, now I don't know the direction in which the reasoning happened, because I suspect that he found it easy to raise things to the power of 4 and get a better fit. But then he went on to think about the physiological meaning of that. And so, I don't know, sorry, I pasted a huge chunk of text here, but basically the insight, this is just to remind me to say this, is the insight was that he thought about this in terms of probability. So you know that when you have conditional probability of event A and event B happening, you multiply the probabilities, right? So he was imagining that there were 4 gating particles. Again, they didn't know about ion channels. So he speculated that there were these ion channel like particles, which all of them needed to be open in order for sodium to flow. And that's where the multiplication came from. So he gave a physical interpretation, which and they were very kind of open to say, you might model this differently, and it might just be an ad hoc fit, but here's one way to think about it. And it turned out to be a really powerful way to think about it that influenced the search for ion channels.
So could we go back to the previous slide? I had a couple of thoughts. So I feel bad that my probabilities it's less accurate than I wish it to be. But doesn't this assume independence between the four gaining particles? Like,
yes.
Right, which is not really the case.
Yeah. Right. So I think, like, I haven't looked at detailed models of ion channels. Karthik, maybe you have. I don't know. But the Hodgkin Huxley model isn't perfect. There are various deviations from real action potentials. And I'm not sure, but I think, and I couldn't actually, I was looking for this, but I couldn't dig it up in time, that the concept of receptor subunits.
Much later, when they actually visualized the voltage-gated sodium channel and voltage-gated potassium channels, I think both were very, very late. I think
90s,
probably early 90s, when they actually had like the high resolution microscopy and also physiology to do the same. What they found was what we now associate as the ball and socket system, which is that that insight which proved useful was that for sodium, because of the way the conductance was measured, they found that there were like 4 units which were more or less alike, which were kind of working in concert with one another. And that's how the N to the 4 kind of came. And the potassium was actually much, much more complicated. The M cube H1, where you had like 3 parts of it which were
with the ions.
Yeah. I'm sorry. The potassium one was with N to the 4. So they had like 4 subunits, and of the protein molecules which were actually involved in the opening and the closing of the ion channel. And the sodium one was the one where they actually had a ball and socket kind of mechanism, where 3 units were kind of working in concert, and then there was another one which was kind of opening the system. In fact, I'll try to find the picture. I remember where they actually showed the different subunits that make up the ion channels, and you will actually see like while it's not perfect, it's like probably the best ideal mathematical representation for that kind of a subunit, and that's what they've captured here. And yeah, it's marvelous that they could just do it with this is basically like the best form of data fitting that you can think of. That's what they've essentially done with N to the 4 and M cube H. And that's kind of what panned out great results when people actually visualize the ion channels.
So you're saying the counting of 4 ends up being correct?
Yes, more or less. So in a sense that if I were to idealize the sodium channel, voltage-gated sodium channel anyways, this is probably the best idealization.
I'm not sure if the number was correct though, but I think the integer, I think relatively low integers was what an insight, and the idea that you needed, because it might be more than 4 subunits. I'm not sure, but maybe it is 4.
No, it is 4 subunits. It is 4 subunits for voltage-gated sodium.
And they are like roughly independent as far as
right.
Like they're independent of each other. They are all equally dependent on the concentration of the whatever the chemical concentrations, but the voltage, but they're independent. Like all the subunits are independent. So like probabilistically, some of them could be open or closed. Like there's they're not directly dependent on each other.
A lot of things that are technically dependent are still kind of independent to the first order. So.
Right. So there's an element of luck, I suppose, but but if you look into the details of their justification for the functions they used, there's a lot of thermodynamics, and so their intuition led them in the right direction. There was probably some luck there, but I think that again, fortune favors the prepared mind.
It's luck only in the sense that they were right about like how there might be some kind of a sub-circuit in the ion channel level. They couldn't say what it was. And we should also be careful that when we actually go and look at voltage gated sodium channel or potassium channel, we are not talking about just the giant squid axon anymore. We are talking about them across species. So for the animal model that they had, this was probably more or less accurate. Of course, now you have a variety of voltage gated sodium channels in different parts of our nervous system, which are not all 4 subunits all the time, but the idea that they are somewhat independent and how they operate, that idea is still true.
Yeah, and this was the part that I was always sort of confused about when I was sort of first taught about this, is where do those powers come from? And very cool to see this. Ram, did you have something you want to share? You're muted, Ram. Oh, is it the
Purvis book? He talks about the channel. I don't know whether you guys can see it.
Sort of, yeah. I've seen some of those kinds of diagrams.
Yeah, so they show that the proteins have like 4 units. You can see all this 4 vertical pieces, but when you go to potassium, you have all kinds of channels. They are not all forming just the 4 units. But it's in the neuroscience book now. Probably it's there in Eric Kandel's book too, but this is in Dale Purvis, and he talks about this equation, n to the power of 4 and the 4 subunits.
Yeah, I mean, there are alpha and beta subunits of the
that's what it talks about.
Proteins. So it's not so, but at least they got the right aspect of it by at least in one sense, where they for the sodium channel they were able to show the alpha and beta subunits, and only one type of potassium channel is what they actually show here. And if you look at people working in different animal models and biophysics models, you will see that they're constantly tweaking these n to the 4 and m cube h parameters in their biophysics, depending on the animal and the kind of sodium or potassium channel they are working with. So that's something that patch clamp people always like tweak and change depending on the kind of sodium and potassium channels they're using. So we shouldn't be too hung up on the n to the 4 or m cube h, but the overall idea that there are these non-linearities because of these independently operating subunits of the ion channels. That's the more important qualitative jump, that they didn't even know about it, but they were able to kind of like intuitively suggest. That's the one of the greatest predictions. Like 40 years later, it took like an electron microscope and 3D conformal geometry and all that stuff to figure out these subunits.
Yeah. Actually,
in fact, you could think of it like as in addition to perfectly modeling or near perfectly modeling the squid axon, it becomes a toolbox. So it gives you recipes for just doing the same fitting in, like, once people were able to record from primates or whichever animal you're recording from, you don't have to reinvent the wheel. You can just use exactly this way of fitting things. So if you've ever worked with Hodgkin-Huxley models and you see these extremely arbitrary looking 1 minus e to the power of something divided by 1 minus e to the power of something else, it all comes from this framework, and each ion channel has its own bizarre properties, and you can do this for calcium channels too. In fact, there's an online textbook, I forget whose it is, but call it the zoo of ion channels.
In fact, it became a cottage industry once these predictions were made to actually find out how different types of ion channels work, and like indirectly, the neurophysiology is what bolstered X-ray crystallography. People don't realize that this rudimentary physiology experiment was the basis for very advanced crystallography. And I think several people won Nobel Prize for X-ray crystallography of ion channels and so on. So it kind of like propelled an entire field at the molecular and cellular level.
So yeah, there's just more stuff that they did. It's amazing. You won't necessarily catch it because anode break is like, it's very old-fashioned terminology and not, but basically they observe, again, the top is the model, the bottom is a squid axon. This you don't just get action potentials by stimulating positively, you can also hyperpolarize the cell. And when you remove the hyperpolarization, you get this rebound firing. So they did that too. I don't have the figures, but they also did the space transmission part, which is a partial differential equation with the second order in X in space, which is quite laborious to calculate. So yeah, this is the summary of this is their summary, which is why they won the Nobel Prize, and it's so wonderfully understated. The way it all is laid out. It's like, we used the voltage clamp and we predicted the quantitative behavior of the model under a variety of conditions that correspond to those in actual experiments. And yeah, then they got the propagated action potential. I didn't talk about it, but they got the impedance change. Well, yeah, the conductance and impedance changes, the inward movement of sodium, outward movement of potassium. They showed the rest of this stuff is actually easier to talk about once you have a model. They showed that there's a threshold for generating the spike, and also a refractory period, which I believe had been already found over the course of the 30s. So basically, the refractory period is when, once you have an action potential, there's a period called the absolute refractory period where you cannot generate another one. And then there's a relative refractory period where you need a much stronger input in order to produce an action potential. And eventually the cell recovers to where it was when it began. So yeah, they also saw subthreshold oscillations. They also predict, again, and I believe this has been verified, that slowly increasing direct currents will not produce an action potential. A little bit like the frog in the boiling water. And they were very open about the stuff that it didn't quite fit. And that's the Hodgkin Huxley model. So the question about how did they calculate this is amazing. So they were going to use the EDSAC computer, which people who've learned the history of computers probably are familiar with that name, at Cambridge, but it was under repair. So Hodgkin decided to compute the solution to a 4-dimensional differential equation by hand. And apparently he started doing these calculations before some of these experiments were done during the war. So his intuition was like, meaning that he was doing this like even before he had the results. So it's quite amazing. And this is the calculator. I found images of it, the type of calculator, the Brunswiga, that actually involves cranking out numbers. And apparently he carried out around 1 million calculations with this thing. So that's one way to do it. Makes you very grateful for computers. So that's most of what I wanted to talk about the Hodgkin Huxley, the model itself, which, it ends with this differential equation system, which then can lead to all kinds of interesting experimentation and generation of new techniques, and also modeling of groups of neurons. But we can pause and look at a single neuron itself as a dynamical system, which is what we've been talking about before. And this is from Izikovich's textbook, which is, I think, really, really good for dynamical systems to start with. So we didn't talk about all the properties of action potential initiation, but basically you need a certain amount of input in order to get it going. And if you don't, you see this capacitive kind of return back to the membrane potential. But you also see you can have a couple of weak pulses that together are enough to cause a spike. And with the right amount of input, constant current input, you'll get periodic action potential, evenly timed. So even though this is in 4 dimensions, I believe Fitzhugh was the first to look at this from the dynamical systems perspective, which had been just sort of a parallel evolution was going on in the same decade, starting more or less with Henri Poincaré. So Poincaré was sort of set this up, and then people started looking at different sorts of properties in dynamical systems. So Fitzhugh in the late 50s started to look into this, and he noted that you could compare some of what the neuron is doing with the van der Pol oscillator. But basically, to start with, you can look at two of the variables, the potassium activation channel and the membrane potential, and think of that as a phase space. It's not the phase space because it doesn't have all four variables, but it behaves analogously in many situations to the two-dimensional phase space of a van der Pol oscillator. So you can see this sort of failed initiation going back to a stable fixed point. So the membrane potential, which is maintained, is a fixed point. And then if you bump the system sufficiently, you enter into a space where you can have a single spike and then back to stability, or you find yourself in a periodic orbit. And if you looked at the Strogatz chapter, you'll note that if you notice this black dot becoming a white dot, there's a theorem that shows that if you do have limit cycles, so limit cycles, we keep talking about them, but we haven't actually defined them carefully. We'll do that next time. But all those neat tools that we use for talking about attractors, repellers, and all of that in linear systems don't completely work. So you don't have like the sort of perfect ability to predict limit cycles. But there's one theorem which shows that if you have a region where you know that the system will tend to enter it, and somewhere inside there's an unstable fixed point where things tend to leave. So you have a repeller, and then it's sort of a donut basically, where inside the donut, the whole of the donut is a repeller, and then around it you have a region which is unknown, and then you know that everywhere on the outer surface of the donut, the trajectories go inwards. There's a theorem which shows that if that situation arises, then you have a limit cycle, meaning that the system will stabilize on a particular cycle. And stability of cycles is very useful and interesting thing, which is why it was studied by radio engineers in the early days. And so that's one thread which we'll touch on next week probably. But basically what that means is that once you have a particular time period of oscillation and even shape, basically, then small perturbations of that might cause small phase displacements, but other than that, you'll come back to this more or less the same shape of oscillation and time period, which, as you would imagine, is really useful in like, technical applications, but it is also the case for periodic stable firing in models of simplifications of the Hodgkin Huxley system. So this is just another look at that same idea, which is plotting the potassium gating variable against voltage. So you can think of it in terms of those predator-prey models and oscillations where the voltage is kind of like the rabbit, and the recovery variable is like the fox, in the sense that it brings everything back down. So the potassium rushing back in, potassium rushing out after the sodium rushes in is part of the process by which the system returns back to quiescence, or back to its original point. So that's what is pulling the system back towards the resting potential, whereas the sodium current is pulling it away from the membrane resting potential. So just as a, to throw this out there, I haven't looked into this in detail, but John Rinzel and colleagues used dynamical system theory to make a prediction, which was then verified in, I believe, a slice preparation. So in a slice, you've preserved some local circuitry, and you can very carefully control inputs and record from it. So they showed that in one class of neurons, the type 2 neurons, you have another sort of counterintuitive effect, a little bit like the rebound excitation, but in the opposite direction. So these are my simulations of what Rinzel and company did. So here you have an input coming in and you have stable firing. But if you time an increase in input just right, you can silence the cell. And the way that they got to this is by looking at the phase plane and saying, this must be possible. I remember showing this to some students, and they're like, how can this be? How can excitation do this? But it's a very sort of fiddly kind of effect in the sense that you have to time it just right. So no one is saying that this is something that the brain necessarily uses as part of signaling. But at the same time, the brain could, in principle, do things like this to silence trains of firing. It kind of puts you in mind of different ways that the spike profile of a cell could be manipulated. And yeah, that's all I really wanted to, I had to talk about today. I thought I would get to the Fitzhugh paper, but this ended up kind of occupying some attention. So, but I'm reading both these books at the moment, and I highly recommend them. They're quite, I think the one on the right is kind of more academic in a way, and more focused on Galvani and the Galvani-Volta debate. So yeah, it's a very brief tour of the Hodgkin and Huxley system, but we can talk about it more now in the remaining time.
This is probably the stuff that I am like the most familiar with / best resource on. A lot of the dynamical, like the more complicated dynamics that you see in already high dimensional systems of ion currents of various sorts. I do want to point out that the stuff about like rebound, the fact that you can inhibit it and get a spike in response, the brain uses that all the time. And I would not be surprised if the brain used the thing that Rinzel found about like excitation at a specific time can actually shut it off. Like the brain really likes taking advantage of these kind of timing mechanisms. If there's an ongoing cycle and there's like a phase where like giving some input produces a particular response, like the brain generally really likes taking advantage of that because it really likes having like multiplex time scales such that it has various cycles that can interact with each other. And I have a lot of thoughts, feelings, excitement around this topic. However, also this was a lot, and I feel like people should probably use this time to ask questions.
Yeah, there's a lot here. So, and yeah, I myself don't fully understand all of it, but it's quite cool.
In fact, I would go further and say that while we can't directly make a connection to something like what John Rinzel has shown at the single neuron level, we might be able to see it at the single neuron level for sure. But I think there is also use to it at the more larger behavioral level. For example, we have this idea in visual systems called as repetition priming and repetition suppression, right? If you keep repeating the same thing at some point, you kind of are bored. So you kind of want to shut down all your activation along those lines. So some mechanism like this at the network level, if not at the single neuron level, is very much possible that you don't want to be simply staring at the same thing and be, stuck there. So repetition suppression is a known phenomenon in psychophysics and the behavioral idea side of it. And people have also kind of measured it in IT and V4 areas. So it's possible that some, maybe not a single neuron mechanism, maybe some kind of a network level mechanism kind of is also operating similarly. So it's quite possible that both repetition priming and repetition suppression could use these kinds of timing issues as the basis for how to shut down or synchronize more neurons. But yeah, it's an open modeling effort.
Reminds me of a phenomenon. Again, if we think about it from the high level, there are these situations where if you play back to what someone is saying back to them, there's a delay at which it will basically silence the person. It makes it virtually impossible to talk. And it's tempting to think, I mean, you can psychologize it and say that you can't really hear what you're hearing too much of what you're saying, but it's such a strange experience that that it almost demands a kind of more mechanistic process. Yeah, there's some sort of self-prediction, and then you're violating it on some level, and generation of subsequent behavior is effectively suppressed. But yeah, you can imagine at the highest level, and we'll probably get there eventually from here with possible applications of dynamical systems at the broad behavioral level, because, and particularly in situations that seem paradoxical, or where you have bifurcation like behavior at some high level, something changes and you get a qualitatively different dynamics.
So given that with the right timing, inhibition can cause a spike and excitation can stop spiking, how much should I let that compromise my general intuition that excitation causes spikes and vice versa?
My opinion is a lot. That I mean, like, unless you're talking about like epilepsy, that in general, things that are, yeah, I mean, other people can weigh in on this, but I would say that like there are a lot of situations in which inhibitory things that lower the voltage are not inhibitory. The whole basal ganglia operates on this principle. There's no excitatory synapses in the basal ganglia, and yet there's plenty of spiking. So yeah, I would say, you know, like if you're planning on engaging with neuroscience, that's an intuition that's not going to help you very far.
In the
Other people can disagree.
Isn't that because there's like a baseline level of spiking that gets interrupted by inhibition, or is it a mechanism like this?
No, most of the, I mean, you know, I did striatum. The firing rate of if most of the cells in striatum is extremely low. There's not really a ton of baseline firing. What's happening is that they are using that rebound mechanism to communicate with each other. So there is, I mean, there's excitation coming in from cortex, but what's going on within the basal ganglia is pretty much entirely cells using rebound inhibition to communicate with each other.
It's more true of the globus pallidus than an SNR than the striatum, because I don't think the globus pallidus receives any excitation. I actually checked it. So some of the internal structures of the basal ganglia circuit only have inhibition, but it is possible, and I think it's likely that at least some of them are intrinsic spiking cells, meaning that without any input they spike. But if cells the striatum tends to be silent unless it receives excitation, but down below that you could have situations where if they're quiescent, then you kind of know that they're under inhibition, and then when they fire, it's because they've been disinhibited. But more, I mean, there's another reason to kind of be careful about inhibition. In my opinion, excitation, it's sort of like the Anna Karenina principle. All excitation is the same, but every inhibition is inhibitory in its own way. So, and the reason I say that, well, first of all, you can model the effect of an inhibitory synapse using the same sort of framework, right? But it's a little bit more passive normally for for GABA A, which is the faster inhibitory signal. So this GABA molecule binds and then it causes a fast inhibition. But then there's a slower one, which is more complicated. But the reversal potentials of these GABAergic currents mean that in some situations, the GABA itself can be excitatory. Mostly that doesn't happen in adults in the central nervous system. In the developing brain, the reversal potential actually changes. So in early stages, GABA is excitatory in the developing brain, in the womb. And there are situations involving not situations, but phenomena in the HPA axis, the hypothalamic pituitary axis that involve excitatory GABA. I think some of those are abnormal, but I don't know whether there are some situations, natural situations, where you have excitatory gamma. So I don't think these are that common in the central nervous system, but it's still something to keep in mind as a possibility. And there are, I like to think of some of these currents as, so there are additional currents, like weird potassium currents and things like that, which and rebounds and things which are produced by additional currents other than the ones that are mentioned here. So some neurons, like pyramidal cells, have this whole arsenal of currents, the zoo of ion channels. And so you can have a situation where there's a voltage dependent current which opens by virtue of being slightly hyperpolarized, and then it sort of kicks in, exciting the cell once the hyperpolarization is lifted. There's a bunch of these kinds of cells, like the low threshold T-type like the calcium channels can do all kinds of things like this. So there's a lot that inhibition can do because of its secondary effects also.
I will say that a better way to think of it often than inhibition causes spiking and whatever, the wrong way. A better way to think about it is that every ion has an intrinsic reversal potential, and the overall concentration of ions in the cell and outside the cell is not going to hugely change. Like there'll be local changes, but it's not going to overall change. The reversal potential of each ion is going to be pretty much constant for that cell and for that area probably. And so everything that the cell can do with ion channels is pulling the voltage towards some reversal potential of some ion. So as long as you know where those points are in like voltage space, then you know that like if this is a channel for this ion, like it's going to be pulling towards that ion's potential somehow. Like the timing is the only thing that's different between channels, and you can get a ton of different things to happen with the timing, but like, yeah, the actual voltages you can go towards are kind of fixed.
Yeah, it's sort of like a tug of war. Each of these channels is trying to pull the membrane's potential towards its resting potential. But you have to be careful with calcium though, because the calcium concentrations are so low that to model them accurately, you need to actually use the concentration, and which is standard. Even there, you can approximate that because it's just more and more epicycles ultimately, depending on how perfectly you want to fit the actual shape of the phenomenon that you're interested in.
Or you can do multiple calcium concentrations like I did within each cell, which is true. Which is true. So there actually is slight discrepancies between concentrations of calcium within the cell.
Which makes complete sense, because if it's lower the concentration, the more you would expect that. And there's also this whole calcium buffering phenomenon, which I don't fully understand, but there's proteins that buffer calcium, and the specific ways in which they buffer calcium vary. And there's a bunch of inhibitory cells which are marked by these different proteins, and no one really knows what the proteins themselves do. So the calcium part of the story is, there's a lot happening there. And a lot of it has to do with learning and plasticity too.
It is still pretty amazing that like all of neurophys is kind of just versions of this Hodgkin Huxley though. I mean, like the tail currents and stuff like that, like all the analysis they do are like, maybe there's slightly newer analysis, but like a lot of the things are like standard neurophys now. It's pretty amazing.
Yep.
I mean, given the fact that we now have like a menagerie of ion channels and calcium gated this, sodium gated that, voltage gated this, invert rectifying, I don't know, like there are like prepared, thousands of ion channels. Gated. Yeah, ligand gated, like thousands of them. It doesn't matter. Like the idea of the basic idea from the whole Hodgkin Huxley of like sodium and potassium like exchanging, going in and out, that in and out idea is in whatever complexity is what's giving you these insane behaviors. And So in that sense, it is still the golden standard in, if not in every field in biology, at least in neuroscience, it's definitely the golden standard when it comes to a model, because even the most advanced biophysics models that people have right now is just a variation of that with maybe one more equation or one more, addition to the current here or there. And it kind of like captures the larger behavior of the system. So, yeah.
Going over this history, you can't help but think that around the middle of the 20th century was sort of the end of a lot of the low-hanging fruit of neuroscience, in the sense that you could ask very clear questions up until that point and then answer them, because the brain was relatively reliable and the phenomena pretty repeatable. But once you go to the network level, things become very noisy. And then when you get to the behavioral level, it's even, well, there's this interesting phenomenon that happens with multi-level phenomena where you have something quite reliable and you have this sea of noisy phenomena, and you reach another level of reliability. It's sort of like renormalization in a way, although I might not be using that term perfectly accurately. But basically there's this sort of sandwich of noisy levels, consolidation of something reliable. And one sort of almost trivial way to think about it is that you can do exact calculations in your mind, right? So whatever noisy phenomena are happening in this mesoscale between the deterministic Hodgkin Huxley neuron and all the different types of noise that can accumulate, somehow get factored out and are completely irrelevant when you're multiplying numbers in your head. Small numbers, not very large ones.
Probably a lot of that is just because the brain is operating in massively parallel. Like nothing is being done with a single neuron.
Johan,
exactly.
I beg to differ. Mr. Huxley did a million calculations, so he seemed to have not really had a problem with very high level
cranking it out. Yeah. So that's an interesting. And
oh, sorry, I was going to say, it mentioned in the chapter though that like that he used basically special calculation techniques that would mutually check errors, so that it's not like he just did it right the first time. It's like it was a whole production to kind of get that kind of reliability out of it.
Yeah, this is a forward-backward kind of version of Euler, so that each time he took a step forward, he would know whether he had made a mistake. Which these are all the things that this Brown guide really kind of shows, and he only came out in 2019. So, and it's great for people learning neuroscience right now to be able to read this kind of thing and not have to bother about the weird conventions that were used earlier.
Something that's kind of implied here, but you didn't quite touch on, even though you touched on a lot of awesome stuff, and what Trinity is really, really awesome, is that those equations can be represented as a circuit.
Yeah, I showed the circuit, but I didn't really pause.
Did you? Okay, I think maybe you went through quickly. I just missed that.
Which really helps the physicists and engineers coming into neuroscience are like, I recognize that. But
and of course you can extend that further later with multi-compartmental models and the cable theory and what's that book? I keep forgetting that book. The insane cable theory book.
The Book of Genesis.
No,
Roll.
Oh, Roll. Not
RYLL, Roll.
Roll. No. Rawls is one idea of it. The guy in Monash, forget his name. He had this insane cable theory book for neural. Rawls is Edmund Rawls is like one part of it, but then this was extended to different types of cables, and like what happens if it's attenuating and myelinated and not myelinated and branching and not branching and all that. So they did a very detailed analysis of that. I'll figure out the name.
Yeah, adding the spatial component makes the calculations even more complicated. But and Huxley did that too for a short distance.
It's interesting.
So there are passive properties, and then over the people have discovered that you can have active meaning, fancy sodium and potassium dynamics in dendrites. So it's not just the axon and axon initial segment and the axon that will show spikes, but even the dendrites. So there's an even larger sense in which a single cell, is itself a complicated network.
The cable theory person who wrote those 2-volume books is Henry Tuckwell. So there was one on pure, like just nonlinear systems, and then what happens if we add noise and stochasticity, and then also have cables and all that. So it's an insane book if people are interested in this kind of models. Henry Tuckwell and his 2 books.
I think I'll stop the recording now and we can just chat.